Characterizing a protein structure is essential for understanding its function. Mass spectrometry has emerged as a powerful tool for this purpose, especially for protein systems that are difficult to study by traditional methods. To study a protein structure by mass spec, specific chemical reactions are performed in solution that encode a protein's structural information into its mass. One particularly effective approach is to use reagents that covalently modify solvent-accessible amino acid side chains. These reactions lead to mass increases that can be localized with residue-level resolution when combined with proteolytic digestion and tandem mass spectrometry. Here we describe the protocols associated with use of diethylpyrocarbonate, or DEPC, as a covalent labeling reagent together with mass spec detection. DEPC is a highly electrophilic molecule capable of labeling up to 30% of the residues in the average protein, thereby providing excellent structural resolution. DEPC has been successfully used together with mass spec to obtain structural information for many different proteins.
Start by preparing a buffered solution of your protein of interest at a concentration in the range of tens of micromolar. We commonly use a 10 millimolar pH 7.4 MOPS buffer. Alternatively, an existing protein solution may be buffer exchanged into MOPS or another buffer if the original sample contains a buffer that has nucleophilic functional groups. It's necessary that the buffer not have nucleophilic functional groups because they will interfere with the DEPC protein reaction. In a different microtube, prepare a stock solution of 100 millimolar DEPC in dry acetonitrile. Preparing the stock solution in dry acetonitrile is necessary to prevent hydrolysis of DEPC. In a new microtube, prepare a solution of 1 molar imidazole in HPLC-grade water. In a new microtube, mix the MOPS buffer and protein solution.